epigenetics becomes a very important player. And when today you talk about the environment is not so simple, and therefore it has concept as an exposome concept. And what we say as an exposome is the exposure to a human body in terms of ecosystem, in the terms of lifestyle, social. We always talk of an environmental exposure, but the social exposure of own household income, the inequality, and others also completely affects your epigenome. Moreover, when you go further, it becomes a complete physiochemical, which we all know and not have been studied much in detail. So all these together defines the epigenome, and therefore that really defines how you are going to express the subset of genes which will be defined during the course of a pathology or different cancers or diseases. And therefore, when you talk of these things, you don't talk only about the extrinsic factor, but majorly we also talk about a lot of intrinsic factors which are harmless and which are really known to change. Then we talk about macrobiome that really refers the biological processes. And these are the different hallmarks of cancers. And now if you look into another concept which has really emerged in 2022 is called the non-mutational epigenetic reprogramming as one of the key hallmarks of cancer. And why so? If you look into all other hallmarks of cancer, they are not only regulated by the mutational status, but majorly they are regulated by the gene expression status. And why this happens, I will be going more deeper into understanding. We have heard a lot of talks on microRNA, chromatin territories. I will be talking more about the histones and how histone proteins play such an important role in defining the cancer epigenome. So generally we all heard about the modification of the histone proteins and which defines the active euchromatin or a heterochromatin organizations. And now we also know that these histone modifications have been very well identified as histone oncomodification. Depending on their increase, which are denoted with a green star or a red star, which shows a decreased pattern, you can have aberrant gene expression, genomic instability, alteration in a DNA repair mechanism, and cell cycle checkpoints. And when these modifications are happening on a genome, what they are really doing, they are changing the complete gene expression profile. And why so it is important when talking the concept of gene-environment interactions. Today, the concept has emerged which is referred as epigenetic priming in terms of the cancer initiation. What we all know, there may be a genetic alteration in a given individual, but everybody doesn't suffer with a disease. Why so? If a mutation is strongly related for developing a disease, like if you talk of APC mutation or a BRCA1, everybody doesn't develop those individual cancers in colon or breast. So then comes the role of an exposure to the environmental factors, which causes epigenetic priming, leading to further mutation and development of a cancer. But if you look for a genome which don't have a single mutation, but a persistent exposure to any kind of a bad environmental factors you are inserting your genome, that results in a priming of a cell. Cells tries to adapt to the change conditions, but with adaptability can only come with some acquiring some of the driver mutation, leading towards the development of a full-blown cancer. So if you look for the environmental factors leading to an epigenetic priming, plays a very important role in driving a cell toward developing a cancer. And the best known example for the clinics, if you see, a normal cell, if you talk of a GIE, gastric stage, those cell lines of the stomach can undergo atrophy, hyperplasia, hypertrophic dysplasia, and metaplasia. All these phases are quite reversible when they are treated at the right time. But these irreversible phases, if chronic irritant persists for a very longer durations, they will result in developing angioplasty. So the early priming effect, which is happening in these factors, persisting for a very long duration, what we are talking about this, leading into a genetic alteration and developing of a neoplastic status. And now why histone? So switching my trigger from cancer to histone biology, because I will be going much deeper in understanding of the histones. Why these histones are so important? If you look at a single cell organism like fungi, I'm just taking an example of a histone which is referred as H2A or H3. You have a single histone genes. There are two genes in the fungi of H2A, and for H3 there is a single histone gene. And as an organism has become multicellular, subset of genes which have increased without a function. Until this, we don't know the function of so many genes till today because they are quite similar in nature. We know some of the function of some of the histone proteins which are emerging from histone H3, like histone H3.1, 3.2, and 3.3. But what are the different functions, we don't know. But if you look for the histones, they have been now classified into two different categories. One is called replication-independent histones, and these probably are generally referred as variants. So if you look for, there are more than 14 variants, and then we have replication-dependent histones, which are synthesized during the S phase of the cells. And if you see the number, in totality, there are more than 50. So if a single cell contains more than 50 different histone genes. So when we talk of a tissue-specific expression, why can't we believe that different histones are and these genes are not the mutation products, they have own independent promoters? Then why can't we believe when we talk of a tissue-specific gene expression, different combination of the histone proteins can give rise to a different nucleosomes. And if they have different nucleosomes, their chromatin organization will be different. And if chromatin organization is different in different cell types, they will have a different gene expression. And that's what we have shown, I'm not talking about today, very nicely. The tissues which originates from ecto, meso, and endoderm have a different histone isoform profiles and different cell types. So just I will talk about the three themes which we really work in context of the histone biology. So there are different research scholars, few have moved out, few are working in the lab. Differential expression and regulation of histone variants on isoform of cancer. This is one of the key themes which we initiated somewhere around in 2004, and which has we have taken up the problems originating from every thesis into the next PhD project. The another problem which we started with, effect of cell cycle dependent chromatin landscape during resistance and DNA damage response. And why this theme was so important? When we talk of a microarray analysis, RNA-seq, or any proteomics analysis, we take the cell lines, but whether those cell lines are in the same stage of cell cycle profile or not will define their RNA as well as the proteomics. If the two cells are in different phases of the cell cycle, their RNA as well as proteome profile will vary drastically. And those will be predicted as a change happening amongst the cell line, but they are not emerging because the profile of a chromatin is different in different phases of the cell cycle. Histone modifications are different, DNA methylation status are different as well as the microRNA status. So therefore we have gone much deeper in understanding this aspect. And the third thing which I will be talking today is a histone post-translation modification and the therapeutic potential in cancer. This story I will be taking ahead in much deeper way today with you. So if you look for, again, when we talk of an environment, how it relates with epigenetics, in between sits the metabolism. Whatever you take inside your body is undergoing a different metabolic pathways. It can be one carbon metabolism, it can be glycolysis or TSA. It produces different substrates, either acetyl-CoA, alpha, SAM, acetylmethylene, and these all acts as a substrate for different histone modification or DNA methylation coming from SAM. So therefore, this is a direct association comes between environment, metabolism, and the epigenetics. And therefore, all the histone modification when we talk about, they act as a bi-substrate reactions within the body. So now going deeper into the gastric cancer as an overview. So again, I am coming to this. It's only 5 to 10% associated with the genetic mutations. The 90 to 95% associated with the environmental factors. What are those? Infections like Helicobacter pylori, smoking or alcoholic factors, salt intake, family history, decrease in vitamin C intake, or nitrosamines. And because in India, the problem of them, the patients with having any GI problems like acidity, they self-treat at home. And therefore, there is a delay in presentation to the clinic. therefore this delay in presentation lead to high relapse rate, high locoregional recurrence, poor response to chemotherapy is a major problem in GI and other, in gastric as well as other GI cancers, and poor survival is very, very less. This overall leads toward a poor prognosis. So these are all the work which I will be sharing in few slides have been published. So when we, at Abbott Memorial Hospital, when the surgery is performed, we nicely know the resection margins. So this is the way of surgery. So what we did a multiple profiling of different histone modifications and the two resection margins along with the tumor tissue samples. And overall, when we looked into, we see a nice observation, a global hyperacetylation is observed. And this project was basically initiated from the bedside, from the clinician. When they are doing a surgical resection, there is a high rate of local recurrence in gastric cancer. So, because the resection margins can vary depending on the site they have a tumor. So when we went into overall global methylation, we could not find a significant alteration, but there was significant increase in phosphorylation. The story which I will not be covering today, the increase in CD10 phosphorylation has been nicely correlated with the resection margin. And when the resection margin is around 4 to 5 centimeter, histopathologically, the tumor tissue, the cut margin is normal. But when we look into the epigenetic markers, till 4 centimeters, the levels of S3-CD10 phosphorylation is similar to the tumor. And therefore, at least this, I'm happy to say this has been implemented routinely in the clinics at TMH. If they get a margin of 5 centimeter, the first seizure goes at the 5 centimeter region. So what is the role of decrease in histone acetylation and whether it has any clinical relevance or not? So epigenomic landscape, we observed global hyperacetylation of histones with condensed chromatin in gastric cancer. So what we observed in a normal scenario, it's open organization and it has a condensed organization in this. So what factors can associate? We looked into the levels by ISC, we find a significant increase in the class 1 HDAC or HDAC1. At transcript, we find a significant increase in few of the samples. But moreover, when we are talking about these are the enzymes. So the levels of the protein or the transcript levels have not of much of importance. The activity of enzyme becomes a key player. We find there is a significant difference in the activity of HDAC, which has increased in the tumor samples with no significant alteration in the activity, suggesting there is a global hyperacetylation is happening because of the enhanced HDAC activity. So when we look at the transcript, we could also find that class 1 HDACs are really overexpressed. Moreover, it nicely correlated with overall survival if you talk of class HDAC1 or HDAC2. So what has happened really in the case of normal versus tumor cells? This is just a cartoon representation of all the data. The nice balance of HDAC is maintained. So the standard organization of chromatin is nicely maintained. But when there is an increase in HDAC activity, you have a highly condensed chromatin. This is what we have nice observation. So what we thought of, the lot of drugs in clinics, they are DNA interacting. If you talk of epigenics, cisplatin, oxaloplatin. So you have a highly condensed chromatin, you throw a DNA interacting drugs, then the binding of the drugs will be less, and the lot of drugs will go into systemic circulation, causing a toxicity. So our approach was, if we treat this condensed chromatin with an HDAC inhibitors, we can open the chromatin organization, leading to enhanced binding of the drug, and if the binding is significantly enhanced, it will lead to more of a cell death, and thereby less of a systemic circulation and less toxicity to the human beings. So with that approach, we started our project. This is just to show the binding of the drugs in 3 different regimes. When you treat the post-treatment of valproic acid, concurrent treatment, pre-treatment, and the post-treatment regime where the HDAC was given accordingly. Whenever the HDAC was given in the pre-treatment regime, which is highlighted in yellow, the binding of the drug in an in vitro experiment was significantly high. suggesting, yes, I can open the chromatin and I can do the binding, which was shown by different experiments. We have shown it by macrofocal nucleus. We have shown by the acetylation statuses because of the time. I'm not going deeper. What we came out, if I treat with an HDAC inhibitor, there's an increased tumor gene expression activity, cell cycle arrest, differentiation, and death happens. So this is what we have shown in the in vitro context in multiple cell lines, but this is just to represent AGS. When you have a pre-treatment regime, you can appreciate there is a significant cell death in response to different HDAC inhibitors, which are clinically approved by FDA. And then when you throw the two key drugs, cisplatin or oxaliplatin, the cell death happens in in vitro condition. But in in vitro condition, we went ahead into a preclinical work, and what we showed, when you treat valproic acid, because why we have taken the valproic, it's a class 1 HDAC inhibitor, and routinely used in clinics till today for treatment of epilepsy, and it has been taken for 30 years along with for pregnant ladies also. It has no side effects. So what we have done, we have treated with valproic acid as a sensitizer, which is an IC20 dose, just to open the chromatin. There was a significant decrease in tumor volume in comparison to other cisplatin, and VPA or control doesn't induce the cell death without any toxicity. So what we came out here, so if we can categorize a patient into two different categories, which has high HDAC or a low HDAC activity, and if you go into clinical trial.gov.in, there are more than 174 clinical trials done in the last 15 to 20 years on HDAC inhibitors. They say it works very well in the liquid tumors, but not in the case of solid tumors. The problem lies where they all have looked into the levels of the transcript. Nobody has looked into the levels of the HDAC activity. And when we correlated our own findings with the levels of the protein or the levels of the transcript with the HDAC activity, only 24 to 30% holds true. So if the patients have a low, high, comes under the category of a low HDAC, and you treat with an HDAC inhibitor, they are going to show the toxicity very high, and therefore statistics will fall back. So this concept we brought into the picture. But the problem was there was no protocol to suggest how to measure the levels of HDAC activity in the serum sample. So we developed a method for that, and therefore now we are able to measure the levels of HDAC activity in the serum samples. And thus, the dissection of serum-based histone modifiers and histone modification became really very helpful to us in defining which patient has to be really treated with the HDAC inhibitor before a chemotherapy and not which to be treated with. And therefore, taking this, all the preclinical work from the laboratory, we have moved into the translational study, and this is which is going on in the human sample to build a proof of concept. Yes, the HDAC. So the patients are treated with the routine chemotherapy treatments, and then we collect the blood sample and try to build up a relationship. The patient with the high HDAC activity are non-responsive. The results are very favorable, and maybe we will start shortly with a phase 1 on this. But the problem here, if you see, we could maintain a tumor, but when these animals were left for around 6 months post-treatment of valproic acid and cisplatin, they came back with the tumor again. So what to do? We all published our data. This was published in gastroenterology. I was very happy. The student got a postdoc based on that. He went abroad. But when we do this experiment further, when we left the animals in animal house for around 3 months, they started coming back with the tumor. So they suggest some is left over there, which is literally non-palpable, and something has to be done for that. So there we started, when we looked into those, that was the new concept came, which is referred as drug-tolerant persistent cells. So these are some 5% of the cells. They survive easily in treatment of a drug, and we refer them as a tolerant persistence. And if we leave this population, they come back and give rise to the, again, a parental population. But if the drug continues further, they develop a resistance and thereby a relapse. So therefore, what was the key importance was to understand what is drug-tolerant persistent cells and with which mechanisms they are surviving. So we prepared those models. We published recently a paper which talked about histone acetylation, a key determinant of acquired cisplatin resistance. If you look at your tumor, what I showed you, it's a condensed chromatin. But the resistant chromatin behave totally different. I will talk about it. We showed by different technologies, RNA-seq and profiling, such as important chromatin-associated changes in the stem cells. And we further went ahead and showed the intake of glucose, higher glycolysis with a large production of acetyl-CoA, which results in a highly open chromatin organization, leading to AKT phosphorylation and further pathways into it. Therefore, earlier we thought of tumor to be treated with an HDAC inhibitor. But the population is resistant, then comes the role of a HAT inhibitor. So the two conditions behave totally different in context of tumor versus the resistant tumors. So what we have at this point of time, in a normal population, we have an open chromatin organization with higher levels of acetyl-CoA. When we go into the tumor population, you have a higher condensed chromatin, higher levels of SAM which favors methylation, lactate increases, acetyl-CoA decreases. In the resistant, the phenomena becomes almost similar to the parental population. But in between sits another population which we refer as drug-tolerant persistent cells. How much time? 10 minutes. So we started looking into the transcriptomic profile of the drug-tolerant parental and the resistant cells, and what we came out that there is a subset of genes which are only enhanced in the drug-tolerant persistent cells. So if you look for, this is the drug-tolerant persistent cell population. This is the cisplatin resistance, this is the parental. If you remember, I have clearly shown you the epigenomic profile of these two populations overlap with each other. Nicely, their transcriptomic profiles also significantly overlaps. However, the DTP population's transcriptomic profile is totally different than the parental or the resistant cells. So this was some of the morphological alteration. We could see they have enlarged nuclei, nucleocytoplasmic ratio is altered, cell size is much bigger. There is an enrichment in the G1 phase of cell cycle, with the higher levels of improved by higher levels of cyclin D1, decrease in cyclin B1, and the K67 is also low, suggesting these cells are non-proliferative and they are lying ideal or in a senescent state in the tumor population. Now to go again back into the chromatin organization, the dynamic of acetylation. The drug-tolerant persistent cells have a low level of acetylation. Resistance, already we have shown it increases. The enzyme are altered, HDAC or the HAT GCL5. This data is shown by confocal microscopy, which matches with the Western blot. Activity, again we look into, the HDAC activity is high, but there is no significant change in the levels of another substrate, which is acetyl-CoA. We went into methylation status, and what we find is a significant increase in H3K9 trimethylation, H3K27 trimethylation, which are heterochromatic mark. There is an enhanced activity of the methyltransferase activity, along with a significant increase in the levels of the SAM, suggesting that one carbon metabolism has become much faster. The levels of the enzymes are also high. So looking into whether, along with the, earlier we have done the simple experiment with HDAC inhibitors, and we have shown they regress the tumor, but not the complete regression is coming, and this is relapsing. So we wanted to see what is happening in the context of the histone methylation inhibitors. Just to cut story short, low-dose treatment with specific inhibitors results in the inhibition of histone mark without apoptosis in parental cells. So we could find out a dose very nicely with all the three inhibitors, which is only IC20, and now we can treat with them without affecting the survival of the cells. So this is just, we are trying to sensitize the cells either in vitro or in vivo context. So we made a treatment regime almost 24 hours before the treatment of cisplatin for 3 rounds of treatment. And when we looked into this, again we found out there is a simply regression of a tumor. But if you look for the number of DTP cells, the drug-tolerant persistent cells which are produced with the inhibitor of methylation is significantly reduced. But the tumor again remains this. When we left these animals again back, They have not come back for a little longer duration of 8 to 9 months, but again it started coming back at a very slow profile, slow rate of slow cell proliferation. So pre-treatment method followed by cisplatin resulted in significant reduction in DTP cells in culture, however no complete response in an in vivo model. So then we came down to the metabolism, because we have seen the production of SAM, again the acetyl-CoA, quickly. When we were doing some electron microscopy studies, we find the mitochondria are highly elongated in these gut-tolerant persistent cells. We did a molecular alternation, we see increase in copy number, the sizes are significantly increased. Then we looked into the fission and fusion markers. We could find increase in the fusion and the fission markers. Mitochondrial biogenesis factors we much into deeper. That also find to increase, the polarity increases, along with the PGB1 alpha, which is the key in the mitochondrial biogenesis, also enhance the DTP. Because this mitochondrial population is giving an advantage for easy cell survival. So if this is happening more there, we find enhanced ROS. If the ROS is there, cell should not survive. If we went more deeper into it, you find the antioxidant activities are also enhanced significantly to take care of the ROS within a cell. We went more into the levels of the metabolites, how they are surviving. We looked into some of the glucose uptake, the pyruvate, the lactate. This is again your phosphoenol pyruvate pathway. So all have increased, suggesting the cells are surviving because of phosphoenol pyruvate. So what we thought of led to the dual treatments. Because the OXPHOS is high, we started including metformin in the system. We looked more into along with the HDAC. I will not go into the in vitro experiment, I just go quickly to the in vivo. Now you will appreciate we have seen a significant decrease in the tumor volume when we are giving VP and cisplatin. The treatment is stopped here, the animal could not develop a tumor again back into the state. And then when we have given this valproic and metformin, they have been given at a very low dose. So if you and all three have been given at a very low dose of the treatment. So that's what we are talking today about, a metronomic chemotherapy. If a patient is treated with a low dose of a chemotherapy, and then if it survives, continuously go on taking as an antibiotic or a paracetamol or any other drug, then at least we can remove the burden of tumor in an individual, which gives a better survival of hospital. So what we came out here in the gut-tolerant persistent cells, this is the highestly condensed organization with significant increase in the methylation mark. We could inhibit OXPHOS as well as HDAC. So I'm here just sitting at the bottom of the